Okay, so then you can also classify neurons by function. And so we have afferent, and we have efferent, and we have association, or interneurons. Afferent mean coming into the brain. I'll try to draw a brain here. That's terrible. But anyway, so there's your brain. So there's your afferent, and your efferent are here. And then your association neurons are the ones that are inside the brain there, and spinal cord. So you can classify them by function. So afferent neurons bring signals to the brain. Stimulus is detected by receptors. They're on dendrites, and then it travels along an axon to the spinal cord where it synapses with a motor neuron or interneuron. And you have either unipolar ones, those are coming from most places, or the special senses are the ones that are bipolar. So you have the most are unipolar. Special senses are the only ones that are bipolar. Then you have, I'll drop down the bottom, the efferent motor neurons. Those are multipolar neurons. Take the nerve impulse out of the central nervous system through the spinal cord or cranial nerves to an effector, which is a muscle or a gland. Then you have association neurons, which are also multipolar, and those are 90% of the neurons that you have in the central nervous system. Okay, so, and the spinal cord, the interneurons can synapse with other chains of neurons to send a signal to the brain, or synapse with a motor neuron. So in the brain, you're having a lot of synapses between a synapse is where two neurons, where there's a gap where either dendrites and an axon come together, or an axon and an effector like a muscle or gland cell comes together. So a lot of the synapses in the brain are between interneurons. You do have motor neurons that come up into the brain and drop off a signal, and those interneurons then send the signal back and forth between themselves and decide what to do. Then they send it. A sensory neuron comes up and drops off a signal, and then the interneurons talk back and forth amongst themselves, then send a signal back out of motor neurons. So, next slide. So here's the functional classification of neurons. So you have sensory neuron bring a signal in, and then you have interneuron, which are only in the central nervous system. Then you have a motor neuron take signal back out. So if sensory neuron is afferent, it's unipolar. Interneuron is multipolar, and a motor neuron is also multipolar. So the association and the efferent neurons are both multipolar. The afferent neurons sensory neurons are unipolar, almost all of them except for the ones that are in the nose and eye. Those are bipolar. Next slide. Okay, so now we have the structural and functional classification of neurons down. The question is, like, where are these guys? We've talked about, you have these sensory neurons come up into the spinal cord, and then you have motor neurons come back out of the spinal cord. But where does all this happen? Well, there's something called the dorsal root ganglia. Okay, and so this is the spinal cord here. Okay, and this is the dorsal side, because you recognize your spinous process here and here, and here's your ventral side here, where the vertebral cell body is, and then you have the spinal cord. So coming in on afferent neuron comes up in here, and then there's this little bulge. There's a little bulge here. Okay, and that's on the dorsal side, and that's the root of the spinal nerve, so it's called the dorsal root ganglion. A ganglion is where ganglion is a cluster of cell bodies. Okay, and they're all linked by synapses. They form a swelling in the nerve. Nerve is a bundle of axons. Okay, so we're going to look at when you look at those pictures of the slides, you'll see the neuron cell body, the nucleus, the processes, and satellite cells all around the motor neuron cell bodies. And then on a cerebral cortex slide, these are what we usually do in the lab. We look at neuron cell body and nucleus and processes and axon and dendrites. on the dorsal ganglion slide, we would see the cluster of cell bodies. So I think there's a slide is one of these next couple ones. Next slide. So no, not yet. So this is a cross section of the spinal cord. Here's your dorsal root ganglion here. Okay. So here's your afferent signal coming in here. There's all the cell bodies are here, all clustered, making this all big and fat. Okay. And then the signal goes on into here, and then it can go up into the brain, and then a signal come back out, and you'll have a signal come back out, and this will be the efferent signal, and this is the afferent signal coming in. So this is sensory, and afferent or efferent is motor, going back out again. So that's why it says over here, sensory nerve coming in, okay, and the motor nerve is going back out. So there's your pathway. Next slide. So here's your dorsal root ganglion. So you have all these clusters of these neuron cell bodies. Here's one here, neuron cell body, another one here, okay, and they have all these cells here, here, there, there, and there. I can't circle all of them, there's too many. So just all those dark dots, those are all the satellite cell bodies. And wrapped around the sensory neuron cell bodies in the dorsal root ganglion. Next slide. And so the reason the dorsal root ganglion is this collection of cell bodies, the reason it's swollen is because those cell bodies are fatter than the axons. So the axons are pretty skinny, the cell bodies are fat, so they get a swelling if you have a cluster of those. Here's another view of the multipolar neuron. Here's the neuron cell body. I showed you this picture before. And here's the axon. So here's to be your axon hillock coming off of here. Here's your axon coming down here. And you see some axon collaterals coming off like that there. And here's some dendrites coming off of here, coming off the neuron cell body, dendrites, and dendrites coming off. Next slide. Okay, myelination of axons. I showed you the myelin sheath. The lipoprotein is the plasma membrane of the cells wrapping around the axon. And the two neuroglial cells that form the myelin sheath, the oligodendrocytes, are in the central nervous system. They wrap their processes around multiple axons. The cell body does not wrap around the axon, so there is no neurolemma in the central nervous system with the oligodendrocytes. The Schwann cells, on the other hand, are in the peripheral nervous system. They wrap around a small section of axon. The whole cell wraps around the axon. There's multiple layers of plasma membrane around it, and the cytomembrane and the cytoplasm and the nucleus are pushed to the outside, to the periphery. It forms what's called the neurolemma, which is the outermost cytoplasmic layer. So I already did the simulation of the Schwann cell wrapping around an axon. That was the plastic bag with the pencil. Next slide. Okay. The myelin sheath is not continuous. Those gaps I showed you, those nodes of Ranvier that are 1 to 2 microns long, micrometers long. The cells are about 100 microns long. So the nodes of Ranvier are 1 to 2 microns. The Schwann cell is about 100 micrometers. Microns is the other word for micrometers in length. So if you have a 1 meter long nerve fiber, a nerve fiber is the axon, then you will have 10,000 Schwann cells along 1 meter, which is about 3 feet long. Okay. You can also have axons that do not have a myelin sheath. They are called unmyelinated. They still have neuroglial cells around them, but it's only a thin layer of Schwann cell or oligodendrocyte plasma membrane around the axon. Unmyelinated axons conduct nerve impulses much more slowly than myelinated axons, or than myelinated fibers, if you want to call axon a fiber. And those are interchangeable words. So infants and young children have less myelin than adults. So infants are slower, less coordinated movements. And also, just nutrition is very important, especially early in development, but all the way through development, through pregnancy. So and infants need more dietary fat for proper nervous system development, because you need fat, you need that lipid going into those plasma membranes to form all those Schwann cells. Because you have 10,000 of those for every single. You may have 10,000 of them for every single axon. So every single neuron is going to need a whole bunch of Schwann cells. Anyway, next slide. So here's a myelinated axon. It has a single Schwann cell wrapped around the axon. It wraps around and around and around the axon. So you can see this is the you see the neural lemma there. So you have just get oriented noted around VA. Here's a Schwann cell. Here's a Schwann cell here. Okay, and then you cut it off here. So here's your axon, and then you have the myelin sheath. Then Schwann cell cytoplasm is all around the outside. That's what this pink area is here, there, and then you have your nucleus there. So the outside part is called the where all the cytoplasm and the nucleus is called the neurolemma. Next slide. And then this is a picture of your lecture text, just showing how the Schwann cell myelinates the axon. It starts wrapping around the axon and continues wrapping. And finally, you get the neurolemma as well as the multiple layers of myelin. And let's see, so here's an axon here, and here's the myelin sheath around it. And this shows the difference between the peripheral nervous system. This is the PNS up here, and this is the CNS down here, because you have oligodendrocyte down here, and you have a Schwann cell up here. So you have an on the bottom. You have oligodendrocytes. Oligodendrocytes can myelinate multiple axons in the central nervous system. Next slide. Here's what an unmyelinated axon would look like. You still have some lipoprotein cell membrane wrapped around the axon, but it's not as much as in a myelinated axon. The Schwann cells aren't wrapped all the way around. They're like only wrapped around maybe once. And a single cell folds around several axons. So the nerve impulse doesn't travel as quickly down these. And so when you send a signal to your muscles, the nerve impulses may arrive at the muscles at different times from different axons, and so your movements will often be uncoordinated. So if you lose myelin, this is what happens in MS and other disorders. If you lose myelin, then the axons are not myelinated as much anymore, and so you lose the speed of transmission in some of those, or I should say the nerve impulse will travel at different speeds in different axons, and so your muscle movements become less coordinated. It becomes much more difficult to coordinate your muscle movements. Okay, next slide. Okay, normally we would observe an axon and a neurolemma and myelin sheath and nose of Braun VA under a microscope, but luckily we do have a picture. So here we go. So here's so you can see your neurolemma. So that's where a little bit of cytoplasm would be. Then you have a myelin sheath. So that's this dark blue part here. All that's myelin sheath there. Then we have a node of Braun VA here. So you see how that little gap forms right there. Goes down in here and here. So that's the two ends. There's a Schwann cell over here and a Schwann cell over here. And they don't touch. They leave a little gap. And then you have your axon, and that's your axon right in the middle here that you can see. So, okay, next slide. Okay, so you have axons, and they're wrapped in Schwann cells in the peripheral nervous system. But people talk about nerves and nerve fibers. So what's a nerve and what's a nerve fiber? Well, a nerve fiber is a single axon wrapped with Schwann cell. Okay, well, Schwann cells. Okay, so you have your Schwann cells. I'm going to draw these. And then you have your axon going down the middle of it, like that. So that's a nerve fiber. You have a single axon with with nerve cells. So if you look at this over here on the right, we have myelinated axon of one neuron, so that would be a nerve fiber. Okay, here would be an unmyelinated axon of one neuron. See, this one doesn't have Schwann cells on it. So you might have, you could mix myelinated and unmyelinated axons within a bundle, and you have bundles of nerve fibers. So each axon is a nerve fiber. Okay, then you can bundle them all into a single bundle, and here's, so I'm going to draw a little circle around that whole thing. So this is called a fascicle. There's the word. It's written for me already. So you can have a fascicle. You have multiple fascicles. So here's one. Then this would be a fascicle. It would be a fascicle. They don't have to be round. They can be misshapen. Here's another fascicle. And here's a fascicle. Here's a fascicle. Here's a fascicle. And here's a fascicle. Yes, I just drew circles around all of them. So, okay. So those are all fascicles. And each of these is wrapped with something. So you have myelination around the axon, and then you have connective tissue that's wrapped around the axon, and that connective tissue is called endoneurium. So you have endoneurium wrapped around each individual axon. Then wrapped around each fascicle, you have perineurium. Then around a bundle of fascicles, so here's your whole bundle here, and that is all wrapped with epineurium. So your whole bundle of fascicles is called a nerve. Okay, and then you have bundles of nerve fibers are called fascicles, and then each nerve fiber itself is wrapped with Schwann cell, which is then wrapped with connective tissue called endoneurium. So if you look on the left here, you can see the epineurium is on the outside here. Perineurium goes around. Here's a bundle of fascicles there. I'm not sure where the edge of it is there. Maybe it probably goes up across here. And then each one of these, I can get that guy out of the way. I guess I got to do it fast, huh? So each individual nerve, I find a good nerve fiber. So each individual nerve fiber, like I've circled several there, but like look at the prominent one in the middle. So that each one of those is a single axon wrapped in endoneurium. So if you look at the endoneurium in this picture, it looks like it's pointing to blue, which you might mistake with the myelination of a Schwann cell, but that's actually supposed to be a different color of blue. That is actually connective tissue that wraps around the Schwann cell, which wraps around the axon. So just to be clear about that. Okay, next slide. Okay, so we're going to move over a little bit, talk about gray matter and white matter in the central nervous system. And so when you think about gray matter and white matter, everybody's heard about gray matter, and everybody, people talk about it occasionally. Using your brain, use your gray matter. Well, where does gray matter and white matter come from? Well, if you look at, here's some stakes, and you have USDA select, USDA choice, and USDA prime. You have lots of marbling. Well, marbling means you have two different colors. You have the white and the red. So the white is fat, and the red is the skeletal muscle. It's the meat. So the white is fat. So fat is white, and so that's what. So we have gray matter and white matter in your central nervous system. So that's what you have. That's what makes the white matter white. The gray matter is gray because it doesn't have as much fat in it. So anyway, so the myelin, and where's the fat come from? Well, the fat is the myelin sheaths that's around all those axons. So you have the phospholipid, lipid meaning fat, plasma membrane. So the myelin sheaths are all made of that phospholipid. And so that makes the myelin sheaths white in color. So if you have just one axon, it's kind of hard to see. But if you have a lot of myelinated axons, the nervous tissue will appear white, a whole big group of it. If you have very few myelinated axons, the nervous tissue will appear gray. So, so the lipoprotein of the plasma membrane of Schwann cells and oligodendrocytes, this gives white matter its color. And white matter are tracts or groups of myelinated axons in the central nervous system. Gray matter are the unmyelinated areas. They consist of neuron cell bodies, so that doesn't have much in the way of lipid around it, except for the plasma membrane around the cell body, but it's not a big group of it. And the dendrites and axon terminals and neuroglia. In the brain, you have outer layer, which is called the cortex. It's gray matter, and the inner layer is white matter. And there are isolated areas of the brain inside their gray matter, and they contain neuron cell bodies as well. The spinal cord is flipped. The outer layer is white matter. The inner layer is gray matter. It's also central and H-shaped. So, and here is, we're going to show pictures of, there's a frontal section of the brain and a transverse section of the spinal cord in the next slides. Next slide. So this is a frontal section of of a cadaver brain showing the white matter and gray matter. And you have gray matter, as you can see, is all around the outside here. Okay, and going this way, and around here, and around here, like that. And you see better here, and then around here, and then around here, and around here. And the white matter is all on the inside here. So this should actually tell you something about the architecture of the brain. So we have the outside, we have these neuron cell bodies. Lots and lots and lots of neuron cell bodies all grouped together. Dendrites and axons are all communicating back and forth, and we have lots and lots and lots of them. And then we have their axons coming out. Axons are all nice and thin. So we have all the axons coming out, and the axons are all going to go somewhere. So the shape of your brain is going to be like this. Have that, and then you have another bulge. It's going to have a whole bunch of neuron cell bodies in it, and axons coming out of it, going down in different tracks. So that's why you have these bulges. Why these each bulge is called a gyrus. These bulges in the brain. So the neuron cell bodies on the outside, and the white matter on the inside is all the axons going leaving the neuron cell bodies and going down to the spinal cord. Next slide. Here's a transverse section of the spinal cord, and what we have is reverse. So we have white matter on the outside and gray matter on the interior. So this is all gray matter. This all gray matter. This stuff. These, and we're going to learn the parts of this, what all these parts are called. But here's all gray matter. So this is all neuron cell bodies. Are all in here. Okay. And so these are all neuron cell bodies all in here. And so you have axons come in, and you have axons go back out again. So you have white matter here, and this is white because the axons are all, these aren't cell bodies, these are actually going up vertically. So I'm doing a cross section through the axons. These are all fibers that are coming straight up. So if your spinal cord is like this, these are all going up. All this white matter are all the axons going up and down. So these are called, so this white matter is actually called white columns. Okay, so that gives you a kind of visual that those are axons going up and down through that white matter. Next slide. Here's just another picture of the spinal cord cross section, transverse section of the spinal cord. WM is white matter. That's white matter. So this is gray matter. Here's all different parts. There's a dorsal horn of the gray matter. There's a ventral horn. There's a lateral horn of gray matter. and there's actually anterior and posterior white columns of axons that are going up and down in the white matter. But the point is that the white matter on the spinal cord is on the outside, whereas in the brain the white matter is on the inside and the gray matter is on the outside. So that's why the cerebral cortex, the cortex being the outside part, is important, because it has all the neuron cell bodies, which are parts that do all the work and communicate back and forth. Next slide.